Well, I had a great time in Columbia, Missouri, and my presentation was well received by the people at the Common Ground Fair, which I look forward to attending to again. Unfortunately, my presentation was not able to be displayed. So, in addition to the in-person conversation, I'd like to make the original presentation available to the people from the Common Ground Fair and anyone else who would benefit from this presentation about the Missouri area in general and the pest dynamics of that place and generally in cannabis. This is the Plainswarm, Pest Dynamics in the Ozark Plainscape. My name is Matthew Gates, founder of Zenthanol Consulting. I've been active for 14 years doing IPM support. I've worked mainly in cannabis at this point, but I have extensive experience in gerbera, roses, lilies. Also, I spent 2 years in China, where I would help with sorghum, tea, and other crops. I have several publications and presentations on IPM in cannabis and other crops as well. I'm very excited about the contributions I made to the New York State Cannabis Sativa Guidebook, working with Cornell Cooperative Extension and other important professionals and experts in the space. I also have a SciComm YouTube channel, which this presentation is going to be uploaded to, called Zenthanol, and I've also got a personal account on X or Instagram at SyncAngel. If you're interested to learn more about cannabis IPM generally, you can check out some of the other publications that I've made or videos that I've made on my YouTube channel that talk about pest and plant dynamics and plant health generally. Usually, I like to start off my presentations talking about this concept that I use called the Everswarm. It's supposed to be a representation of the various kinds of pests that you're going to be dealing with, as well as their enablers, both environmental enablers, like dynamics like temperature, humidity, things like this, as well as the aggregated various pests you're going to deal with that are constantly changing. They're constantly evolving spatio-temporally, so across space and time. There's a few examples here. We have the beet leafhopper, an insect that on its own doesn't really do very much damage. Indeed, most leafhoppers don't do much damage, but this one in particular carries beet curlytop virus, an incredibly virulent virus that is found in many different crops, including cannabis, and it's going to continue to be found, especially because BCTV can't actually be cured. The only thing we can do is prevent the vector, which is the leafhopper itself. So in this way, it's important to know that the beet leafhopper might not be a problem, but it may carry, and not all of them do, carry this pathogen. Other insects like aphids also carry lots of pathogens, but they also have symbionts that we should pay attention to. The pea aphid is an insect with many different bacteria important for various functions, but one of them in particular is Hamiltonella defensa, which not only by itself affects some kinds of parasites that would infect aphids, but some strains have various populations of a virus, a bacteriophage, which normally would infect bacteria like Hamiltonella defensa, but in fact is what we call lysogenic. It actually is encoded into the genome, the genetic code of the bacteria. This virus is called APSC, which stands for Acethrosiphon pisum, which is the species name of the pea aphid, secondary endosymbiont, APSE. So when you're using biocontrols, the combination of these microbes can really inhibit the efficacy of what would normally be a really sustainable effect. Budworm moths have a similar capability with their gut bacteria, like Enterococcus. So the insect by itself is very damaging, but these bacteria, specifically those two, among others, can disrupt the capabilities of things like Bacillus thuringiensis or BT and the toxins that they produce, rendering, again, a very crucial, very critical treatment basically ineffectual, as the various plants and microbiota that are important for the crops that we're worried about, as well as the human element, the humans that are affecting and augmenting the way that the crop system develops and grows, versus the pest system, not just insects, but mites, fungi, bacteria, viruses, and other enablers. This Everswarm concept acknowledges a multi-domain context where we can have changes between regions, seasons, adaptations in various populations, and invasion capabilities. One pest is simple, but 40 plus pests at the same time, simultaneously, consecutively, whatever, are of course much more complex. It's not like a Bruce Lee flick where one pest becomes a problem by itself, isolated, you defeat it, the next pest comes around, you defeat it in sequential lockstep. You can often have the case where you have many different pests and other contributing factors that make the pest issue more deleterious or more common, and also inhibits your ability to treat it. And that's the reality rather than the hope that people might have, which is a lot less untenable. While the evolutionary progress of the Everswarm cannot really be stopped, I would say it can be attenuated pretty well, but it depends on the resources that people have access to. I advise that people analyze and disrupt three particular targets: PED, pest physiology, pest ecology, and pest development. Their capabilities and vulnerabilities so you know what to target. Their establishment and range, which can often be found out so that you can understand where they do well, where they might not do so well, and how that's relevant to where you are particularly, and also their development, research on adaptations, selection pressures, and things that shape their survivability. Section A of this slide shows the interaction between a host microbiome, in this case a plant, and a pathogen or mutualist microbiome. So the pathogen or symbiont and the host have their direct interactions, but we have on the host side, we have eukaryotic microbes like fungi and protists and other sorts of microbes, viruses like bacteriophages that are found in the soil and also on leaves, much more commonly than you'd think, and prokaryotic microbes like archaea, which are often not talked about, and bacteria as well. A similar sort of thing is occurring for the pathogen or symbiont, potentially, where viruses, eukaryotic microbes, and prokaryotic microbes are interacting with each other and the pathogen, and then also secondarily affecting the interaction between the pathogen and the host. For a more concrete example, we have the oomycete in figure 1 that is deploying antimicrobial products. These products, which are depicted as green, red, brown, and purple structures, manipulate the phylosphere of the microbiota, which are the blue rods, which you might think of as bacteria or something like this. And so by disrupting the phytoplane, the surface of the plant foliage, the pathogen can severely disrupt what would normally be a more inhospitable environment for itself. B and C of the slide shows a parasitic microbe, an oomycete, being aided by a typically mutualistic microbe of the plant by changing the way that the stomata, which are important for respiration in the leaves, open and close, allowing the infection of the oomycete to be much more effective. But alone, you wouldn't expect the mutualist to be a problem, and this is why IPM can be really complicated, and aspects of IPM are very fuzzy without quantification. Things like plant resistance really are based and predicated on not just genetic capabilities, but also the microbiota, and also how at a genetic level the plant interacts not only with its parasites, but also with its mutualists. I often like to depict IPM as a holistic system of systems approach, a multi-domain strategy, where you want optimization at all levels, and you also want to be able to anticipate adaptation factors. Figure 1 on the right shows research outreach for IPM, but I think it's also incredibly important for us as cultivators to understand some of the dynamics that are not only true for other crops, but of course cannabis as well. One aspect is information outreach, where researchers synthesize information, make observations, publish research, and that research is then hopefully utilized by growers, the general public, to make informed decisions on what to vote for or what they're comfortable with, and also for other researchers to then mount onto that information more information and have this snowballing effect. There's also a communal approval and action effect. Obviously, regulations influence IPM, and so do the norms and traditions of various societies and communities, and they're going to be very different. Missouri, of course, is going to be different than California or Michigan or Florida. And there's, of course, a sustainability aspect. Environmental consideration is paramount. More and more people are becoming more savvy about how these systems operate, what they're comfortable with, what they're willing to tell their constituents and their representatives that they're not comfortable with, is all a very important aspect of, as the figure depicts, research outreach for IPM, but also IPM generally in your operations therein. This slide depicts very neatly the invasion threat of different countries around the world in 4 different contexts. A shows the overall invasion threat. Lighter blue colors show a higher severity, while darker blue colors show a lower severity. For A, the overall invasion threat is highest in the USA and Canada, parts of South America like Chile, Argentina, and Paraguay, parts of southern Africa, such as Malawi and Madagascar. We have parts of Europe, such as Norway, Finland, Denmark, Bosnia, Bulgaria, Ukraine, Switzerland, the Czech Republic, and Albania, among others. In Asia, there is Korea, Vietnam, Laos, Cambodia, Bangladesh, and Nepal. And Meganesia, the area that we would consider to encompass, for example, Australia, which is depicted here, as well as New Zealand and related land masses, are also affected. B depicts total invasion cost in millions of US dollars. We can see here that China has a particularly high level compared to the other countries. In C, we have total invasion cost as a factor of GDP percent, and in some of the countries where even the invasion is very high, like the USA, the total invasion cost as a GDP fraction is actually very low, whereas in other countries, like Madagascar, for example, invasion threat level is high overall, and the cost is going to be very severe as well. And while Missouri is nestled snugly into the midwestern heart of the United States of America, trade does happen in the USA between these countries, and it's important to recognize that some of these countries may become important vectors for pests that we care about, especially in cannabis. As you can see here, one Connex box in 421 days went 75,762 kilometers by ship, 5,197 kilometers by train, and 2,171 kilometers by road. It went through the US, down into South America, across the Cape of Africa, all up and down Southeastern Asia, went through Western Asia, up into Europe, into the UK, and back again. It reached many countries where the merchandise imports reached billions or trillions of USD, and it's very likely that with all of the various examples of Connex boxes, the hundreds of thousands of them that are moved through nautical, aerial, and ground transport, these changes are likely to continue. The vectors for pests are also likely to increase, and it's a reality that pests will adapt in these various regions, become stronger, and then move to other locations where these adaptations may significantly benefit their invasion capability. In addition to natural flyways, many pests, whether they're fungi as a spore or insects that have a flying life stage, are able to move on aerial currents that can actually go great distances. They are also aided by the trade. A lot of times things will move from, as this depiction shows, Central and Southern America to Europe, Southeast and Eastern Asia, and even parts of Africa. And we also see a lot of movement of especially agricultural and ornamental and other sort of plant-based trade that moves from Central and Southern America into India, Southeast Asia, East Asia, and even, again, parts of Africa. This is significant because in like 500 million years, when we had Pangea and other sorts of supercontinents, it still would have taken longer back then when the continents were together than it would now for pests to move great distances or across the earth, essentially. It's the fastest, longest, and safest movement of plants and pests in Earth's history, and again, it's going to continue. We can see the movement of container freight and air freight has steadily increased since 1992, and it shows no sign of not continuing. And trade agreements have basically skyrocketed since the 1990s, and we also shouldn't expect that to change very much either. Finally, there's good evidence to support that alien species records, which are the documentations of invasive pests and other organisms, are sharply correlated with imports as a factor of GDP. Since the first quarter of the 1800s, we can see that alien species records and imports move up somewhat steadily until the early 1900s, where they sharply drop. No points for guessing why that was, all the way into the 1950s, where again it starts to rise, and rise much sharper than it was previously. So, at a strategic level, IPM of cannabis and other crops must be locally focused, sustainable. It has to be varied so that we don't rely on one selection pressure to which pests will be able to adapt. It has to be rapidly deployable, and it has to be communal. In other words, it has to be useful for as many people as possible, and it also can't inflict a detriment on others, and it has to be communal, meaning that it can't benefit only one group. It should benefit as many people as possible and not inflict detrimental effects on others at the benefit of a single group or several groups. In other words, we need to have a multi-layered resilience to pests. Otherwise, the pests will deploy their resilience factors to great effect instead. So this presentation happened in Columbia, Missouri, at what I like to call the Ozark Prairie threshold. When I was looking at the ecoregion of the area, this is smack dab in the middle of two very different regions. And we can see that Missouri, more generally, is kind of at the border of this northeastern pest pocket that this diagram shows, where the number of invasive forest insects and pathogens per county in the US increases a lot on the west coast, but also in the sort of northern central region, especially in the upper northeastern area. Columbia, Missouri seemed to be around 18, a little bit less than 22 pests per county itself. So a recent research report came out that detailed a survey of common pests in cannabis for the United States. And they divided it up into several regions, and Missouri is kind of at this sort of nexus point between the other regions, this sort of north-central region. Now, across the results, we can see that for microbial pests, fungi were the obvious dominating force, with oomycetes, which are very similar to fungi and operate very similarly, the secondary group. For Missouri, in the north-central region, its total for fungi pests was 83%, first of all regions, and the major ones were Botrytis, and probably other Budrot pathogens as well, powdery mildew, Fusarium, and Septoria, which I've been hearing about a lot and having to deal with a lot lately. About 12% were oomycetes, and the two big ones were Pythium and Phytophthora, which are extremely common agricultural pests in general. Very, very virulent. For arthropod pests, which are like insects and mites and their kin, we have, for the north central region, about 23% being moths, again, first of all regions, but tied. And we have the corn earworm, the European corn borer, the fall armyworm, the hemp borer, and the stalk borer being the major species that were found and reported in the survey. About 15 plus 8% or 23% were what we call bugs, so in the group Hemiptera. Now I say 15 plus 8% because for some reason they seem to um, separate aphids from other Hemiptera, and I'm not sure why they did that because typically they are combined as a group, but you can see that specifically 8%, so almost 10% were specifically aphids. So that's interesting. So that's cannabis aphid, western flower thrips, which are not actually Hemiptera, they're in the close relative, the Thyssanotera, and stink bugs, various species, along with ligas and leafhoppers. Here you can see figure 2 of the survey that shows the percent of occurrence and the number of instances. So for all 5 regions we have red, 4 is yellow, 3 is blue, and 2 regions is green. We should probably expect the lower instances in various regions to maybe increase as the survey numbers increase as well. Pythium and Phytophthora were the 2 biggest ones in all 5 regions. Bacterial leaf spot was also extremely high in all 5. And we also have beak curlytop, hop latent viroid, and some other viruses as well, topping the microbial groups. We have Botrytis, powdery mildew, Cercospora leaf spot, which looks very similar to Septoria, or can at least, Fusarium, two different kinds of Fusarium infections, though perhaps the same species or even different kinds of Fusarium species. Because this is diseases of pests, we should understand that a disease is the sort of physiological effect, the symptoms of the pest, but the actual pest organism has a species and a designation like that. Some other big ones include bipolaris or zonate leaf spot, septoria, fusarium of the head, which is also something we're seeing a lot more in Kentucky. We've got sclerotinia, which is also another necrotroph, kind of like botrytis, and has a similar profile or can. Anthracnose is really common in other crops. Rhizoctonia is a common root pathogen as well, also found in all 5 regions, about 40% of occurrences. These higher occurrences for like botrytis and powdery mildew is close and exceeding to 60 and 70%. So the Common Ground Fair is smack dab in the regional nexus of the temperate prairies and like the Ozark Highlands areas, which is designated here 9.2.4 and 8.4.5 respectively. We were right in the middle of Kansas City and St. Louis, which obviously has an effect on the movement of pests through trade and that kind of a thing, as there was a pretty big highway system, and these are 2 polar cities that sometimes have a bit of a rivalry, right? Other ecoregions to consider are the Central Irregular Plains, the Western Corn Belt Plains, which is where we're going to see some of the flatland and some of the long distance moving pests that can get onto the winds. But we also have the woodlands and the highlands of Ozark area, and also the interior river valleys and hills region as well, which kind of actually caps the Ozark area when we consider specifically the proximity it has to Common Ground Fair. So as a little area report, if you were to kind of consider this region at a strategic level, we have the red dot is the Common Ground Fair area as a mock-up example. I would consider the major cities and landscapes, of course. Big consideration is the I-70 for pest movement, major cities going east-west and west-east, and the interstate itself being also where other state travel is going to merge through. The Mark Twain National Forest is also going to be probably a major um, source for various insects because of the nature of these forest woodlands. They can have a lot of life and probably don't need me to tell you that it's going to be quite lush. But an interesting thing to note is that some of the surface territory is so ancient and hasn't been like swallowed up by other land or whatever that it dates back to the Precambrian period, which is like way over 500 million years ago. Pretty incredible to consider that this surface of land has kind of stayed at the surface for so long. But there's also a corn belt influence, which I designate with this purple arrow. showing the movement from like basically the corn belt, where a lot of times budworms are going to be found. So for people growing indoor, this might be less relevant, but for people growing outdoor in greenhouses, I've definitely had, and people in the audience definitely remarked on their interactions with budworms, also aphids for that matter. More locally, we can also see again the 124, the East Highway 124, that people kind of turned off to get onto the road, and the various purple arrows show roadways by which that you can get traffic. And I know that people have to often in Missouri move great distances and have to stock up and things like this. So although that can kind of suppress movement a little bit, it can also increase the chance for infection if something is contaminated and it gets stockpiled somewhere. You can have more of an eruption from that area, if that makes sense. I'd also consider the lake cluster around here. This selection of like water bodies could also be, and likely is, more biodiverse, and there's various insects, both predators, but also maybe pests and pathogens that could also sort of bed down essentially in the area and then kind of constantly move out, especially when we consider how often spores will travel such great distances. And I know that humidity can increase quite a bit, which makes things really good for fungi and other microbial pathogens. And here's a satellite image of the landscape as well. I will say that when you look at it, the lakes, because they're also somewhat green in the same color as the trees, it can be hard to pick them out a little bit, but I find that moving and looking at your area with this kind of technological tool can be very illustrative of some of the basic things that maybe you haven't considered before. So if I were to set up at the common ground area as if it were sort of a grow space, I might suggest to somebody to put sensors or traps somewhere around the perimeter and maybe some areas internally. And I might recommend the tree lines here, both because they're a natural barrier, but also because their density, especially in this central location that we have here, could be more useful and more likely to pick up things. Not only do you want to be picking up pests, but you also want to be picking up potentially predators. You might also get parasitoid wasps and other things that can actually be really helpful for you to understand if other things that you're doing to help out, like maybe growing certain kinds of attractants for predators and parasites and things like this, banker plants and things like that. But also, if you want to establish other sorts of organisms or other sorts of interactions, then it would be helpful to know that these things are actually increasing, especially if you have data before doing it and after doing it. I would also bias my sensors and other sorts of traps, whether they glue traps or pheromone traps, like for budworms, for example, towards the roadways as well. And I would have specific ones that were closest to the roadway just to be sure. So here we have three examples, the blue arrows here. And we can also depict the various sort of metaphorical thrust or salience into the area. Most proximately interior river valley area, the Ozark highlands coming up from the south, and then we also have the central irregular plains coming in from the north area. Powdery mildew is a really common pest in cannabis, and I got a lot of questions about it at the Common Ground Fair. Generally speaking, these would be Golovinomyces species and others in the family Erysiphiaceae. One thing I like to note about powdery mildew that basically I don't think anyone else really talks about is an interesting aspect of its lineage. So on the left here we have what's called a phylogenetic tree. It basically just shows the relative similarity and the relationship that different groups of fungi have with each other in this case. You can see that a large section has been outlined with the Helotiales, so that's an order of fungi. Here you can see I've outlined in green the Erysiphoid clade, which is just a group, and we have two family names. We have the Arachnopezzaceae and the Erysiphaceae. Now, it's a funny name, but arachno means spider, and pezes means puff. So it's the spider puff family, which is a little bit easier to say. and I might encourage people to use it more often. And we have the Ericifaceae, which are the powdery mildews. They have some interesting similarities to beneficial soil fungi, especially detrivores. They have shared cellulase genes, the Ericifaceae, the powdery mildews, and the Arachnopezaceae. They have a shared communication pathways, what are called MLO or mildew locus O genes, and both mycorrhizae and powdery mildews fuse with the plant cell membrane upon colonization, which is really interesting because that takes a very sophisticated ability to communicate with the plant cell. And there's other interesting things to note there as well. In cannabis, we have two tribes mainly, although the taxonomy of powdery mildew is very complicated and often changes, so this could change too. But generally, we have the Golovinomycetae, of which the genus Golovinomyces is important. Golovinomyces siceracerum or lettuce powdery mildew is found in cannabis. And we also have Golovinomyces ambrosiae as well, which I like to call the immortal powdery mildew, but it doesn't really have a common name. That name being because ambrosiae refers to the immortals, not because it's impossible to deal with. Then we have a different tribe, Cystotheceae, of which Podosphaera has been found in cannabis. And if you're interested to learn more, I have denoted some of the research reports that these diagrams come from, and I often try to color coordinate so you can tell which texts are coming from which claims, just to be upfront and allow people to make their own decisions and look at the research in a more detailed way. So why is powdery mildew a huge threat? Well, it's a common threat. It's also the fact that powdery mildews, their genome is about 4 times the size of other molds or ascomycetes. And most of the size has to do with transposable elements, which are jumping genes that oftentimes have genes related to their ability to infect the plants. So these change often, and those mutations sometimes become lemons, and other times lead to incredibly virulent strains, essentially. And when you're making all these mutations per spore, and you're making like hundreds of thousands of spores, and there's several that are going to be not only in your area, on your plants maybe, but also those sources being from natural areas, hosts that are not cannabis but are in the area, can really make it so that you're constantly having to deal with this, I don't know, Everswarm of some kind. So it's helpful to know that although a lot of times spores don't go too far, they certainly can go far, over 700 kilometers. And when they sexually reproduce, they create chasmothecia, which resist things like heat, cold, dryness, and also wet weather. Spores also in powdery mildew will store a lot of water, unlike other kinds of fungal spores, which allows them to germinate in dry conditions. And I have a video on my channel that goes over some of the powdery mildews that have traveled from North America to Asia and the Western Eurasian steppe. This is a common byway for powdery mildews to move between North America and Asia, and also Europe for that matter. So we should expect this to also happen more. And when we consider cannabis cultivation increasing, some of these regions will have different dynamics as well. Every spore is a dirole, I like to say, because powdery mildews are what we call obligate biotrophic parasites. That just means that they must have a host, and that host has to be living tissue, unlike molds that you find on bread and things like that. Powdery mildew also, like I said earlier, tends to mutate really rapidly, and the successful asexual spores will reproduce. They make a bunch of volume. Those that are unsuccessful is whatever. They just have to be ready once. They have to win once. The cultivator has to win every time, essentially. And your pest gets the vote. You also might be dealing with things that might make your plants more susceptible. So that can also be, of course, a complicating factor. I mentioned virulence factors before. That's just basically things like enzymes, toxins, and other kinds of kinetic forces like what we call their haustoria or their appressorium that penetrates through the plant cell. So they use enzymes and toxins to break down the plant cell, and then they penetrate it with the force of basically the air pressure of like large truck tires. Actually, it's pretty hard. It's like a couple megapascals of force um, in like a needle point at a microscopic level. This is what powdery mildews look like for people who have not ever encountered them. A shows a fledgling colony in a typical circular form with a lot of sort of mealy lines or sort of powdery structures. We can see as A goes to D, the colonies try to expand, and they also recolonize the same tissues. They are not systemic, however. Unlike other fungi like Botrytis and Fusarium, they are basically only on the epidermis of the plant. Although it's often not lethal, it's a problem, obviously, and when it affects your plants and also gets onto your flower, which is not typical in my experience, but it can happen. And I have a thought that perhaps different species may have different capabilities or different sort of profiles and preferences. We see this with other fungi and other powdery mildews, so it's not out of the realm of possibility to expect that to be at least plausible here. But how does it work? Well, in this case, we have a conidiospore, an asexual spore. It hits the plant surface. It produces a structure called aprosorium. Here in the diagram, we can see it actually has gone to a guard cell, so a stomata cell where respiration happens in the plant. So it has chosen wisely. It produces this little patch that will penetrate into the plant cell. Then it will produce a thing called a hostorium about 12 to 14 hours past infection. Now here's a critical point. If the plant cell was able to not only detect the movement, the spore and the hypha, but also the movement into the plant cell, if it was able to exhaust the powdery mildew at this point, then the infection might totally be destroyed and resistance is assured. However, if the powdery mildew is able to basically inoculate the plant cell with a bunch of commands, genetic and otherwise, with chemicals and proteins that suppress the immune system and also cause the plant cell to act in a way that's more favorable for the powdery mildew with resource acquisition and things like this, then we will move on to the production of secondary hyphae and also the other plant cells being infected and the production of more spores. So I mentioned MLO genes before. There are different kinds of MLO genes as depicted here in the diagram. Some are related to reproduction, some are related to powdery mildew infection, some are related to symbiotic fungal colonization, and some are related to what's called root thigmotropism, which is the detection of like pressure by the plant cells in the roots. So this is important because basically, the long story short, is that these MLO genes are susceptibility genes. They allow powdery mildew to colonize the plant tissue. However, if these genes are removed, or some of these genes are basically not present through a mutation or something, then the powdery mildew infection basically becomes totally inhibited. Almost totally. Usually, often, in other crops, totally. But if that's the case, then also mycorrhizae and other beneficial fungi interactions are also inhibited because it's the plant cell's reaction to pressure, the thigmotropism, that allows both powdery mildew and mycorrhizal fungi to be accepted because the plant cell changes in a way that makes it receptive if the right signals and other sorts of things are emitted. So powdery mildews know how to exploit that pathway, which also lends credit to their potential as a beneficial soil fungus in a time before now, because they adapted to not only break down plant cells, which were probably dead, but then they were able to move on to communicating and bypassing the immune system of a living plant host. So from a good guy to a bad guy, essentially. So this is some of those effects that I was talking about. On the right of this diagram, we have an incompatible interaction where the powdery mildew is inhibited by papilla, which are basically like, think of them like, you know how like maybe you have a scar. A scar is made up of tissues, and those tissues are not like the regular tissues. They're basically an amalgamation of a bunch of structural components that keep everything kind of secure. And that's what plant cells essentially produce, is this sort of hardened scar tissue that keeps the physical penetration of the powdery mildew from happening. And essentially, they have to be able to detect the powdery mildew before the colonization attempt really gets underway. And if they're able to do that, then they're able to mass defense and this effects. The other thing that can happen is that if it gets through and it produces this hostorium, which itself produces effectors that will change the genetic expression of the plant cell in various ways. The plant cell has the capability of producing these what are called MVBs that physically latch onto the structure and suffocate it, basically. Keep it from receiving resources, keep it from influencing the plant cell. It's at this point that the plant cell is able to destroy the hostorium or inhibit it greatly. And this will depend on the virulence of the powdery mildew and this sort of spear and shield relationship. Unfortunately, even if the latter thing happens, the powdery mildew probably was still able to not only suppress the plant cell, but also suppress cells nearby, but was also able to produce the spores and probably colonize other parts of the plant that locally have not benefited from the global immune response that happens in the first several hours of infection. Here's an example of wheat powdery mildew, which does not colonize cannabis to my knowledge, and is often considered to be very specific or produce specialized strains of wheat infection. But we have an example of a gene that is found in a lot of powdery mildews, this sort of sterol 14 dimethylase gene, or CYP51. And those that have it in various different ways, different mutations, this copy number variation they're talking about here, they have different kinds of ability to resist pesticides and also plant toxins and things like that. This gene produces a protein that degrades various toxins. And one of those toxins is microbutanil, which you may know as Eagle 20. And so those with the lighter color and higher copy number variation, they are more likely to be resistant because they have a more varied defense profile. Those that are low in copy number variation are more susceptible, actually, to these toxins like microbutanil. And you can see that this map shows a change across Europe and parts of Africa and the Middle Eastern Peninsula. You can see that there's a great amount of variation in some locations and low variation in others. And we can expect this to be the case probably for other powdery mildews that might even have changes with these genes. We just haven't seen it or researched it yet. You can also see that things have changed pretty significantly between the different sampling times. 1980 to 2001, 2007 to 2020 is mostly higher copy number variation in comparison to before, but now we see many more isolates. Before we saw between 0 to 20. Now we're seeing between 0 and as much as 60 copy number variation isolates, and many of them are of the higher copy number variation. So we can see that resistance is increasing across the regions, but we do have some places that are less resistant than others. A different gene also makes the resistance or susceptibility to microbutanil and other toxins more tenable, or less tenable. Here we have the haplotype Y165F and V295L. I know, really stunning naming here by the biologists, but you can see with the color coordination that those that have these different basically genetic groups are more or less resistant. Those that are closer to yellow and red are more susceptible. So we can see a great increased susceptibility in like the central region of Europe, but we can see that resistance has increased a lot in some pretty agriculturally intense regions like the south of Spain, Portugal, parts of Turkey, and also Italy for that matter. So if you were living in these areas, if you were dealing with wheat powdery mildew, you would probably react appropriately by trying to hedge your bets and maybe not rely on certain compounds or even, unfortunately, certain resistance traits that the plants might have that have now been bypassed by this change in genetic spread. So a little history lesson about powdery mildew. Essentially, powdery mildews originate about 100 million years ago. Their first hosts were tree hosts that were in the sort of polar forests about, again, 100 million years ago, the world looked more like this. There was a lot more water that was encroaching on land masses. South America and Africa were much closer. Um, Parts of Europe and North America and the Arctic were a lot closer in, and so we saw greater spread of animals and plants. So this original range went from the polar forest down into more equatorial regions until about 50 million years ago, where we saw also at the same time aster host movement coming up. So as this is happening between 100 and 50 million years, these tree hosts move down. 83 million years ago or so, the asters developed in the southern tip of what is now South America, and they spread either into North America and then the Eurasian continent area and Africa, or in a different pattern, but they did spread. They spread between North America, Europe, and Africa between 50 and 40 million years ago, at the same time that these powdery mildew tree hosts were moving down. And when they found each other, powdery mildews, some of them had mutations that allowed them to eventually colonize these asters, which eventually became basically the most successful flowering plant species group ever. And to this day, it's still true, which of course propelled powdery mildews into many other plants that we deal with right now for food, for ornamental pleasure, for horticultural science, for all these other important avenues of not only the ecology, but also of human understanding. And so powdery mildews have become a huge pest because many of these flowering plants are important crops for us. They're important to us for understanding ecological health as well. The next pathogen that we have here is Botrytis and other bud rot pathogens, and there are other species. And you can see a depiction of colonization here from A to F. You can see that, at least in this case, the infection started in a fan leaf. It moved and necrotized the tissue. The necrotized tissue is brown, and chlorosis follows with the yellowing of this green leaf that eventually moved into the bud. The floral tissue itself started to necrotize and brown, and then you can see tufts of white that is forming, which becomes the bud rot that we know about, which are the spores and other external hyphae. So botrytis should be appreciated because not only does it come from an important family of fungi, the Sclerotiniaceae, but the botrytis species group, of which there are 40, is actually kind of usually pretty specialized. It's only Botrytis cinerea that colonizes over 1,400 plant species. It's a black sheep of the group, and it can exist both asymptomatically in this host as well as symptomatically as we know it to be. It can also move between parent to offspring, what we call vertical transmission. Botrytis, unlike powdery mildew, is systemic. We call its behavior biphasic. It goes from a lifestyle where it basically coexists with the plant to a lifestyle where it is actively destroying the plant. And several fungi also colonize only floral tissues that can be called bud rot. Botrytis' name comes from the Greek botrus, which means a cluster, specifically of grapes, or a grape-like cluster, you might say, and itis, which means disease. And cinerea comes from cinereus, which means ashy in Latin. So basically, ashy cluster, or gray cluster, gray mold. Botrytis has been estimated to cause about $10 to $100 billion USD in agricultural damage per year globally, so it's a big problem for many, and we still haven't been able to crack it in some ways. Botrytis cinerea has also been shown to associate even with marine macroalgae. 6 endophytic strains found in knapweed itself kind of illustrate this point that botrytis is a highly adaptable sort of group, and if we don't recognize that, then we're really doing ourselves a disservice. How does it infect? Well, it eats death. Powdery mildews colonize the epidermal cells like we see in the left diagram, but in the right diagram we can see the typical necrotroph infection. This is Sclerotinia sclerotiorum, but botrytis infects very similarly. It produces a spore. The spore infects the cell. It produces these effectors, like the powdery mildew does, that isolate the cells, suppress the immune system of surrounding cells. It actually manipulates the plant cell to destroy itself using a system that would normally be used against things like viruses, where the plant cell destroying itself would actually inhibit the infection of other plant cells. It would also send signals to other plant cells, which can allow the plant to outrun the pathogen. This infection is illustrated really well in this diagram. I feel like there are various effectors, which you can see at the bottom. We have plant mRNA, which eventually becomes DNA. These are signals. These are genetic, basically, effectors that are going to change how the plant cell expresses its genes. Fungi like Botrytis will also produce small RNAs that will inhibit or otherwise stall the expression of these genes, or maybe even alter them. Variously colored circles, squares, and star patterns are representing the secreted fungal effectors, which will create, for example, the signaling cascade that leads to plant cell death, or PCD. And if that wasn't enough, they can also influence the influx of calcium ions, which is also going to induce plant cell death, the autolysis of the plant cell. We have many compounds that are named after botrytis, which illustrates its importance in plant pathology. We have botritiol, botriendiol, dihydrobotritiol, botrioic acid, epibotritiol, botsinin. So these are all compounds that are toxins, essentially, and manipulators of the plant cell immune system. But we have microbial biocontrols. We have 3 examples here from this research report: Trichoderma asperellum, Bacillus amyloliquefactions, and Gliocladium catenulatum. These 3 have been shown to have a much better effect for the flower compared to the green A control here in diagram on the right. B, C, and D are all color coordinated with the microbes, and we can see that in the graph, compared to the control, the incidence of bud rot was a lot better when applied 2 days before inoculation. These microbial biocontrols do different things, so it can be helpful sometimes to rotate them. Some of them are going to produce antifungal compounds. Some of them are going to parasitize the fungus itself. The violet and red letters show the parasitic profiles, and the blue, orange, and yellow letters show antimicrobial compounds that were inhibiting bud rot production. There's also mycoviruses out there. Here we can see an example of different strains of mycoviruses that were found in China, actually, that inhibit greatly botrytis colonization by inhibiting their ability to produce what is, like powdery mildew, a infective sort of appressorium, or like infection cushion, as they say. So the infection cushion couldn't be produced, but if there was a wound, the botrytis was still able to colonize the leaves. In this case, we can see that the difference here is C and D, where on the right we have the control, the HB Tom 459. Mycoviruses are pretty abundant, and it seems like they're another aspect of holistic ecology that has been left out of the conversation. For understandable reasons, we didn't have good ways of detecting it until now, or even appreciating their multivariate ecological effects. They actually are already being used in the biocontrol of chestnut blight, which is a huge issue in North America. We probably never again will have the forests of chestnut that we had pre-human colonization, and particularly the last 200 years or so, which is very unfortunate, but we might be able to do something of a restorative effect. The mycoviruses have a variable natural effect and distribution. We can see, though, that across a 900,000 square meter area, that these 2 viruses, Botrytis cinerea hypovirus 1 and Botrytis fusari virus 1, were found 29.6% and 14.3% respectively in this population of samples for botrytis. So probably it's a lot more common than we think, and it might even be a factor that we're not even giving credit to when we're building some of the ecological systems and intentionally sustainable systems that we make already. Another big pest in Missouri and other parts of the Midwest are budworms, Helicoverpazea specifically, and also other Heliothineae, which we will get into. Not only their evolution, but also how to mitigate them. We can see here on the left an example of a budworm caterpillar that's pretty far along, coming out of a tunnel. We can see on the bottom left a Helicoverpa moth that is drinking nectar out of a flower. They have a pretty nondescript body, but you can see that they're pretty hardy moths. They do have some girth to them, so they are noticeable if you are able to find them out and about. although usually this will not be active at daytime. And you can also see the very small white eggs of a budworm moth. Different species have basically the same size and coloration for their eggs, but as you can see at the bottom right, the coloration and structure of the budworms will change between life stages, instars as we call them, different caterpillar stages, and also different species. A common control would be Bacillus thuringiensis, which is a bacteria that produces toxins like cry toxins that are very specific to caterpillar biology. Essentially, they create a toxin that causes sepsis. It creates holes in the intestinal lining, which allows the fusion between the hemolymph, which is their blood, and the gut, which is generally pretty lethal. However, the mortality rate will change between the size of the caterpillar and the amount of BT that is being used. So it's important to know how much is being applied and when it's being applied, perhaps even more prudently. But how does it work exactly? These diagrams show in the upper left and the bottom right how what we call toxins, these toxin crystals, these delta endotoxins, they can only be activated in a highly alkaline midgut. So our pH and the pH of many other animals are not compatible. But if they are compatible, if they are at the right pH, the toxins change, the proteins become formed in a different way, and they move to create pores in the cells, which we can see at the upper left and the bottom right. Now, as these various proteins come together, they create what's called cadherin. This cadherin structure creates these pores, and they just start to dot the various tissues of the intestine. There are other proteins besides cry, like CYP, VIP, site, surface layer, and perisporin, and various BT strains will produce different kinds. There's also BT that affects flies, but those BT strains will also not affect caterpillars, so you have to know which ones you're dealing with. Cotton bollworm, which is Helicoverpa armigera, is able to mate with Helicoverpa as well. So these two budworms are kind of synonymous in some ways. Essentially for us, they can both probably feed on cannabis, and they definitely interbreed as well. It's hard to differentiate them by vision, and so in a lot of cases it's important to actually have sequence data to support the conclusion that you have one species over another. But in my experience, it's the corn earworm that I encounter the most, and it makes sense given the propensity of corn in the USA. But cotton bollworm, like corn earworm, have a high chemical resistance profile. Cotton bollworm in particular has been found to be resistant to not only Bt cryproteins, but also DDT, pyrethroids, spinosins, carbamates, organophosphates, which should not be applied in cannabis generally. Carbamate, organophosphates, DDT, and pyrethroids are really not good. But even if you were unscrupulous and wanted to use them, apparently they wouldn't even be all too effective, and you're just going to create a greater selection profile for not only the proteins that affect these, but also the plant toxins that you might care about in more resistant cultivars as they develop. They're also attracted to a lot of the chemical compounds in cannabis specifically. I've highlighted on the right here alpha-pinene, beta-pinene, beta-myrcene, beta-ocimene, limonene, linalool, alpha-farnesene, borneol, beta-caryophyllene, and methyl salicylate are all compounds that are found in cannabis that these budworms are attracted to. And the moths will actually try to locate chemical signals that are similar to the plants that they were raised on. And with the other propensity of ditchweed being so common, I would not be surprised if there's actually been like 200 years of sort of selection for some populations to be more adept at feeding on and resisting the effects of basically cannabis phytochemistry. In China, there is a huge epidemic of resistance for budworms that we can see here in these graphs. Basically, higher expression of certain genes or lower expression of certain genes that are related to toxin resistance. So different expression profiles are associated with different pesticide exposures. Abamectin, chlorantraniliprole, tebufenazide, indoxacarb, chlorpyrifos, lambda-cyhalothrin, dimethyl sulfoxide. All of these are, again, not really products that should be used in cannabis, but it illustrates the point that even these extremely noxious compounds are being resisted, in some places quite readily. We can see that in comparison, North China, which is a huge agricultural megacenter, the usage of pesticides peaked 70 trillion, an area of 10 million hectares, 8 applications per year, whereas in Xinjiang we have 18 tons of pesticide use, over 6 million hectares of area, only 4 applications per year. So there's a big selection difference between these two regions. Somebody did a massive genome sequence of the genes related to adaptation in budworms. The researchers found a major amount, 18,455 genes related to toxin processing, which is important for plant defense resistance, and plant sugar processing, which is important for cold resistance. For example, the T397 mutation of a gene called TRET1 alters the structure of a receptor in the cell wall of the budworm, and it increases the storage of their blood sugar, essentially, called trehalose. These that more rapidly and readily stored these sugars actually benefited almost as a cryoprotectant, because this sugar is inert and does not create ice crystals in the same way that other sugars would when the caterpillar goes to pupate under the soil, and they can survive at minus 20 degrees Celsius, which is pretty incredible. And it may represent the movement of these budworms to other regions, including parts of the USA, where you would normally expect more of a reset, like above the 40th or 42nd parallel or latitude of North America, and other regions for that matter. They've also got an interesting gut microbiome. Enterococcus and Enterobacter neutralize these Bt toxins that we often rely on by increasing the gut alkalinity beyond structural tolerance. Now, the pH of the gut of caterpillars is actually highly alkaline and almost the same magnitude as bleach. Almost, not quite there. So we should appreciate that the gut that they've developed over time degrades and detoxifies many compounds simply by their pH being really, really massively high. Other bacteria that are in budworms include things like Commensalibacter, Chryseobacterium, Acinetobacter, Lactococcus, Apibacter, Bartonella, Isaiah, Orbus, Disconomonas, Morganella, Pseudomonas, and also some Rhizobiales, which are associated with nitrogen-fixing bacteria in plants, which is kind of interesting. Some of the effects that these bacteria have include things like multi-drug resistance, especially Enterobacter, Enterococcus, and Ralstonia, which were found in the guts of budworms. Another research study found that some of these bacteria have various effects or are associated with certain effects. Enterobacter, Enterococcus, Ralstonia, and Acinetobacter were all associated with multi-drug resistance in hospitals, which means that they probably have a lot of toxin degrading compounds that they produce. It is interesting to speculate whether or not some of these specific species, which were also found in hospitals, might also somehow get into caterpillars, into budworms, or also for that matter other insects, and how this might change the game for resistance management, not only from compounds but again natural plant defenses. Delphtia are also known to degrade many toxic substances. Cellulomonas degrades indigestible cellulose, so that can have an effect on maybe sugar production or the digestion of other toxins. So maybe even targeting the symbionts may disrupt budworm development, and the way to do this would maybe to be introduce bacteriophages, viruses that colonize bacteria, ostensibly not only those that would colonize the budworm itself, but those that might target the bacteria specifically, or maybe other gut symbionts for that matter. It's also possible that some antibacterial or antifungal compounds produced by beneficials might also be able to upset the epidermis of the plant cell and maybe also make it harder for bacteria that would help the gut of the budworm from establishing in the caterpillar somehow. Although it has been found that a lot of these bacteria are passed from moth to caterpillar, especially when it's been found that the budworms will feed on the eggshells in order as a probiotic to help their digestion and also colonize their gut with some of these bacteria too. Bt toxin resistance from the People's Republic of China has spread to several countries. We can see here that this Helicoverpa armigera tetraspan 1 or HAT-Tspan 1 mediates cellular signaling. Mutation L315 specifically in this gene confers resistance to the Bt protein toxin Cry1AC, so a specific manifestation of this Cry toxin. So it impairs the ability for the toxin to have the structural damaging effect that I illustrated earlier, which is not great for us. We should also be aware that at a strategic level, we have research that has shown us a lot of information about the behavior and evolution and development of budworms and other insects for that matter. It allows us to forecast and think about what the future holds for us, even if regionally the changes have not expanded yet. We should consider the likelihood of them expanding. As we saw earlier, trade has increased, will increase, and so will the exposure of these pests. In a 600,000 square kilometer region, annual metrics showed in North China through radar and other sampling abilities, 9.3 trillion individuals, 15.5 billion insects per kilometer, and 15,000 tons of biomass annually, which were mainly moths, bugs like aphids, and flies. So this kind of sampling informs at a regional level what you're likely to find, and also tells us that it's likely that some of these resistances and these genetic mutations will probably pass to populations in other regions as well. We know that TRET1 is associated with trihalo storage, it acts as a cryoprotectant, and it allows the survivability of minus 20 degree conditions. This change also is importantly affected by the behavior of the moth. It feels the effects of the change in season from hot to cold. It creates a change in the physiology of the moth, passing these signals of seasonal change to their offspring so that they can have these benefits. Some of the novel avenues of control for budworms and other moths include things like the use of drones. For example, the PATS-C and PATS-X system were used to not only detect through IR, but also kinetically affect the budworm moth. They can be deployed in greenhouses and deployed probably externally as well. And they can also show the movement of various pests in the greenhouse space. So you can get a visual, you could even just scan for the moths, even if you didn't have the drone itself, and understand their detection. Also, what was interesting, particularly for this system, is that it had a 3D model that showed the counts, it showed the regional area in the crop space, and it even showed the nighttime movement in a 3D space. So this kind of sophisticated technology could allow people to maximize their control. And while I don't think that moth guts moving around and onto the flower of cannabis is particularly great, I do think that there are ways to deploy it that makes it more tenable. The last pests that we're going to talk about are aphids, cannabis and rice root aphid being a big one that people talked about at the common fairground. And although there's not a lot of research on cannabis aphids as much, there's much more research on rice root aphids. Fortunately, there are a lot of similarities in control, and there's a few basics to understand as well. The big one that I'd like to pass on is that, much like the other resistances we were talking about, aphid physiology is really robust, and other Hemiptera in general, thrips that a lot of people were encountering in the surveys, also have a really good way of defeating plant toxins, and also doing things like, as you can see in the right, what's called deimatic behavior, which is supposed to frighten things like parasitic wasps and predators that might otherwise maybe make a feast of them. But there are microbes, gut symbionts in particular, that provide immunological benefits, protection from various toxins, and also there might even be secretions that will influence the development and growth of the plant host. A big aspect of the microbial gut symbionts is the digestion of their sugars from phloem. Aphids are basically a living tap that is constantly feeding on plant sugars and high Brix sap. They actually like more sap sugar than less, and they will feed more and for longer periods on high Brix plants, which is interesting because I sometimes hear people say that it's the reverse, but research hasn't really shown that to be the case, and neither has my personal experience. So a lot of these gut enzymes that are produced by bacteria in aphids are very important because otherwise the aphid would not be able to essentially feed on the sugars. Crucially, they also provide essential amino acids that are not really found in plant sap, so without these gut microbes, they would surely not be able to feed on sap. So it's a basically 200 million year tradition at this point. Interestingly, aphids have been shown to have sort of a aphid microbe feedback loop with the soil microbiome. Because they produce sugars, those sugars will get onto the ground eventually or onto the foliage, and that will shape and change the microbial communities. Obviously, there's things like sooty mold that people encounter with honeydew. That's usually black coloration, and it's not really a plant parasite. However, it does mess up the photosynthesis of the tissues that are affected and covered, and it can also lead to a level of senescence and weakness, which can invite the movement of other pathogens into the plant. So although not directly parasitic, it does still have negative effects. The influx of sugars has a major effect on the composition of the microbiome, and in some cases can even be found to create a sort of competition between other microbial species since sugar resources go up. Some microbes make use of it better, other microbes make use of it worse. It upsets some of a balance. Maybe it's beneficial sometimes, maybe it's detrimental other times, but it will change things. Like I said earlier, aphids have Hamiltonella defensa, and they have APSC. On the right, we can see APSC1 is a lysogenic bacteriophage, which means that it usually colonizes the genome of the bacteria itself, and then as the genome gets reproduced and the production of more bacteria happens, the virus itself gets transmitted at the same time. And so little strings of genetic code that the virus provides in the genome of the bacteria allow it to produce a bunch of toxins that affects the colonization of the parasitic wasp. The bacteria does not always have the same effect, however. Sometimes it's more effective, sometimes it's less effective. But it could be a reason why biocontrols like these are not effective in some cases. We can see here that in some cases the protection is 100%. Sometimes the protection is around 50%. And there's also different viral strains too. So as you can see, APSC strains vary wildly, and so the protection offered to aphids vary wildly, but there's selection pressure, of course, because the virus plus bacteria plus aphid system that survives and then clonally reproduces is going to outpace the ones that do not, where parasitoid wasp selection pressure is most important, like, for example, a cultivation area. Now, one thing you can do is attract more biocontrols like parasitic wasps and things like this. This example with the woolly apple aphid wasp that does not colonize cannabis but does show one of the benefits is that when the wasps were provided with not just water or not just honeydew, but honeydew and water or a sugar substance, they were 5 times as longevitous, and it increased their ability to colonize. They produced more eggs, they killed more aphids. They're essentially way more useful if they're properly outfitted. So you should support these biocontrols as much as you can, and there's some niche ways that you can do that by providing banker plants. Sometimes people provide water sources as well. It just depends on your context. Now, one thing that we do find also that it's very hard to tell in the field, but has been shown many times in research, is that there are hyperparasitic wasps. If you can believe it, there are parasitoids that colonize parasitoids that colonize aphids. On the left, we can see different species, Aphis spiracola, Brachycottus, Helichrysy, and Mysis persici. And then we have the hyperparasitoids in the red and yellow colorations. and they are associated with different aphid species that might be used either as a banker for parasitic wasps, like if you have a plant, that plant has an aphid that doesn't feed on your crop but can survive on the host, then the parasitic wasps can come in, colonize those aphids, and then also hopefully colonize, if they're the compatible species, the ones that affect your crop, in this case, cannabis aphid and rice root aphid, some of which would include things like different Aphidius species like Aphidius metricariae that are often found from commercial biocontrol suppliers. So as banker crops increase, you might get more natural enemies, but you also might get more enemies of natural enemies. So it's something to consider. Aphids suppress the plant immune system. We can see that in these GIF images here. On the left, every time you see a bolt of red or yellow coloration, that is the aphid inserting its mouth part into the tissue, and the requisite sort of calcium defense signaling changes. Blue coloration is low calcium, and yellow and red is comparatively high defense signaling associated with that penetration, and also the detection of the saliva and other constituents. On the right, we have an example of a suppressed response on the top, where the aphid was able to suppress the immune response in the plant. On the bottom, however, we see a plant that has been bioprimed. In other words, the immune response has been induced artificially, either by the use of a plant hormone or the expression of a gene constitutively, which can happen in some plant mutations, or secretions produced by certain microbes, either in the soil or on the plant tissues, that primes the plant to change the immune expression profile. In this case, it's really, really ardent, and you can see that the aphid would probably have a lot more time with a bioprimed plant. It would be reduced in its reproduction, it might even not be able to feed very well, and essentially that would make not only the detection, but also the treatment of the aphid population a lot easier. Finally, a bit about viruses that don't get a lot of credit. This research report showed an example of viruses on leaves, viral particles per leaf. Each one of these color globs show a different group of viruses, a different order, which is pretty high in the taxonomic rank, so very different orders, very different virus groups for each color: pink, blue, light blue, brown, dark green, and light green. The estimated minimum viral particles per leaf was 2 million viral particles per leaf, most of which were bacteriophages, which affect bacteria. So again, this concept that there's this sword and shield effect happening between viruses and microbes on the phytoplane of the plant, the surface, as well as in the soil, is an important one. It was found in another research report that both crop and forest foliage had similar levels of viruses, about 10,000 to 600,000 virions per gram of tissue. And what would happen is that the viruses would actually move into the soil from rain and other sorts of factors, and then they'd be kicked up with the dust, and they would travel on the dust and air currents to get onto the other foliage. So this interaction is happening constantly, and perhaps it could lead to an avenue where we would be able to affect negative bacteria and the bacteria that are affecting the insects and other organisms that we're concerned about, in a multi-tiered way that would resist the adaptation strongly to any one point in a multi-point treatment program. Thanks again. I hope that was illustrative of the various pests that we have to deal with in cannabis, the unique paradigms in the Ozark Temperate Plains area of Missouri, and maybe this has inspired a better respect for some of the pests we have to deal with, their strengths, but also their crippling vulnerabilities that with more time and experience we'll be able to exploit with greater vigor as the cannabis community becomes more supported by research and by the experience of great growers.